Right now, your eyes are performing a miracle. In the time it takes to read this sentence, they have captured millions of data points, adjusted to the light, tracked moving text, and sent a perfect image to your brain. The human eye can distinguish 10 million different colors. It can detect a single photon of light in complete darkness. It adjusts its sensitivity across a range of 100 billion to 1. No camera ever built comes close to matching it. The eye is the most sophisticated optical instrument in the known universe. Welcome to the complete science of the human eye, how it works, what makes it extraordinary, and why it is, by any measure, a miracle of biological engineering. Let's start with the basics. What is the eye doing? Its job sounds simple: detect light and send information to the brain. But the mechanism is anything but simple. The eye is roughly spherical, about 24 millimeters in diameter, the size of a small marble. Inside this tiny sphere, one of the most complex biological structures ever evolved: the cornea, the clear dome at the front of the eye. This is where vision actually begins. The cornea does about 70% of the eye's focusing work. The cornea has no blood vessels. This is deliberate. Blood vessels would block light. Instead, the cornea gets its oxygen directly from the air and its nutrients from the fluid behind it, the aqueous humor. Behind the cornea sits a clear fluid that maintains the eye's pressure, nourishes the cornea and lens, and is constantly produced and drained. The iris, the colored part of the eye. Its color is determined by the amount of melanin pigment. Blue eyes, less melanin. Brown eyes, more melanin. Green, somewhere in between. But the iris is not just decorative. It is a muscle, actually two muscles working together, that constantly adjusts the size of the pupil to control how much light enters the eye. The pupil can change size from 2 millimeters in bright light to 8 millimeters in darkness. That's 16 times the area, happening in less than a second. The lens. Behind the iris sits the crystalline lens. Unlike the cornea, the lens can change shape, making it flexible enough to focus on objects at any distance. This focusing process is called accommodation. When you look from your phone to a distant tree, your lens changes shape in milliseconds. You don't feel it, it just happens. The lens is made of transparent proteins called crystallins. These proteins are incredibly long-lived. Some of them are as old as you are. The lens proteins you have now were made when you were a developing fetus. Why does vision blur with age? Over decades, these ancient proteins gradually lose their flexibility. The lens stiffens, accommodation becomes harder. This is presbyopia, the inevitable focusing difficulty of middle age. The vitreous humor. Behind the lens, filling most of the eye's volume, is a clear gel, the vitreous humor. 99% water, held in structure by a network of collagen fibers, floaters. Those transparent shapes drifting across your vision, they're shadows cast by clumps of collagen in the vitreous humor. Completely normal. You're seeing your own eye's interior. Now the retina. This is where the magic truly happens. A thin layer of tissue, just 0.5 millimeters thick, lining the back of the eye. It contains over 130 million light-detecting cells. There are two types of photoreceptor cells in the retina: rods and cones. They are physically different shapes, and they do completely different jobs. Rods, 120 million of them, sensitive to dim light. They work in darkness and low light. They can detect a single photon, but they see only in black and white. Cones, only 6 million, but they give us color and fine detail. They need bright light to work, and they're concentrated in one extraordinary spot, the fovea. Three types of cone cells: S cones sensitive to blue light, M cones sensitive to green, L cones sensitive to red. Every color you see is a combination of signals from these three types. Color blindness, when one or more cone types is absent or malfunctioning. Red-green color blindness, the most common, means the M and L cones respond similarly, making those colors indistinguishable. The fovea, a tiny pit in the center of the retina, only 1.5 millimeters across, but packed with 50,000 tightly clustered cones. This is where you see most clearly. When you focus on something, you're pointing your fovea at it. Your eyes are constantly making tiny movements, microsaccades, to keep the image on the fovea, the blind spot. Every eye has a point with absolutely no photoreceptors, where the optic nerve exits the eye. You have a hole in your vision right now. You just can't see it. Why don't you notice the blind spot? Because your brain fills it in. It uses surrounding information to predict what should be there and paints it in. Your brain is constantly hallucinating your blind spot. Phototransduction, the process by which light becomes a nerve signal. This is the core miracle. How does a photon of light become an electrical message the brain can read? Inside each photoreceptor is a molecule called rhodopsin in rods or photopsin in cones. When a photon hits this molecule, it physically changes shape. That shape change triggers the cascade. One photon, one rhodopsin molecule changing shape, triggers a cascade that amplifies the signal 1 million times. That's why you can see in near total darkness. After the photon is absorbed, the rhodopsin molecule must be reset, regenerated, before it can detect another photon. This takes time. This is why you're temporarily blind when you go from bright light to darkness. The optic nerve. Once photoreceptors generate signals, they pass through two more cell layers, bipolar cells then ganglion cells, before leaving the eye via the optic nerve. Wait, the photoreceptors are at the back of the retina. Light has to pass through all the other layers to reach them. Isn't that backwards? It's backwards by mammalian standards. But it works because the inner layers are largely transparent, and it allows photoreceptors to be in direct contact with the retinal pigment epithelium, the layer that recycles visual pigments and provides nutrients. The optic nerve carries about 1.2 million nerve fibers from each eye. These fibers converge at the optic chiasm, where left and right visual information crosses and splits. At the optic chiasm, the nasal half of each retina's fibers cross over to the opposite side of the brain. The temporal half stays on the same side. This is how each brain hemisphere receives a complete view of its side of the world. The visual cortex. At the back of the brain, one of the most complex structures in neuroscience, it takes up roughly 30% of the entire cortex, more than any other sense. The visual cortex processes visual information in parallel streams. The what stream identifies objects. The where stream tracks location and guides movement, both happening simultaneously. The eye and brain together process visual information in about 150 milliseconds, but you experience it as instantaneous. Your brain backdates the perception to make it feel immediate. Binocular vision. Two eyes, slightly different positions, each sees the world from a slightly different angle. The brain compares the two images to calculate depth. Cover one eye, your depth perception drops significantly. You can still judge depth from monocular cues, size, overlap, shadow, but the precision of binocular depth vanishes. Saccades. Your eyes don't move smoothly across a scene. They make rapid, jerky jumps between fixation points, called saccades, up to 500 times per minute. During a saccade, you are technically blind. The brain suppresses visual processing while the eye moves, otherwise you'd see a dizzying blur every time your eyes shifted. This is called saccadic suppression, peripheral vision. Outside the fovea, cone density drops rapidly, but rod density increases. The periphery is specialized for motion detection, not detail. Your peripheral vision can detect movement that your central vision misses entirely. This is an evolutionary adaptation. In the ancient environment, movement in the periphery meant predator, tears. The eye's surface is protected and nourished by a three-layer tear film. Oil layer on top prevents evaporation, watery layer in middle, main volume, mucin layer at bottom, helps it stick to the eye. You blink about 15 to 20 times per minute. Each blink spreads and replenishes the tear film. You don't notice blinking because the brain suppresses the darkness, the same saccadic suppression mechanism. Emotional tears contain different chemicals than reflex tears or lubricating tears. Emotional tears have higher concentrations of stress hormones. They literally flush stress hormones out of your body. The eye is self-repairing. The cornea can heal a scratch within 24 to 48 hours, one of the fastest healing rates in the body. Corneal stem cells at the edge constantly regenerate the surface. The eye barely moves in its socket, only 50 degrees in each direction. But within that small range, the 6 extraocular muscles coordinate with extraordinary precision. No two people move their eyes in exactly the same pattern. Color constancy. The eye and brain together maintain the perception of stable colors even when the lighting changes dramatically. A white page in candlelight still looks white, even though the light reaching your eye is orange. Edge detection. The retina doesn't just send raw light data to the brain, it pre-processes the image, specifically enhancing edges and contrast through a process called lateral inhibition. Night vision. In darkness, the eye shifts almost entirely to rods, but your sharpest night vision is not straight ahead, it's slightly off-center, because the fovea has no rods. The eye's oxygen demand is extraordinary. The fovea has the highest metabolic rate of any tissue in the human body, higher even than brain neurons firing at full speed. Macular degeneration. When the blood supply to the fovea degrades, central vision disappears while peripheral vision remains. The most common cause of blindness in the developed world. The eye evolved independently at least 40 separate times in the history of life on Earth. The compound eye of an insect, the mirror eye of a scallop, the pinhole eye of a nautilus, the camera eye of humans, all evolved separately. All solve the same problem, detecting light. The fact that the eye evolved independently so many times is extraordinary evidence. Seeing is so important for survival that evolution arrived at optical solutions again and again across completely different lineages. Right now, as you read this, 130 million photoreceptors are firing. Billions of calculations are happening in your visual cortex. Your brain is constructing the world you experience, not capturing it, but actively building it. A sphere 24 millimeters across, 130 million light-detecting cells, amplification of 1 million times, 10 million colors, 1.2 million nerve fibers to the brain, processing in 150 milliseconds. The human eye is not just an optical instrument, it is the window through which consciousness meets the world.